Hyderabad campus. So she'll be sharing with us all those things. So please be seated, ma'am. Please be seated. She'll keep
Welcome to all the participants, all the guests on the dais. So today I'll be presenting a topic which is upon ecology, regeneration potential and conservation of the freshwater planarians from Pune, Maharashtra. So giving a just brief introduction to you about the planaria is because we have the basic idea which belongs to just the 9th standard textbook or whatever we have learned in the bachelor's, that there is a planaria, it is a flatworm belonging to the Platyhelminthes phylum, and it is having a tremendous regeneration potential. That is what is the basic idea which belongs to all the zoologists, but this is not the whole component which you all know. There is a lot more which is there in the planarian the phylum of planaria. And its class, it is very diverse to explore, and the research arena is very well establishing right now.
Just for your information of the juries, so each presentation has 100 marks, of which 20 marks will be given, like out of 20 marks, whatever mark you to give on innovation and creativity on the topic. 15 marks has been kept for scientific merit. 15 marks have been kept for impact potential. 15 marks have been kept for technical implementation. 10 marks have been kept for interdisciplinary integration. 10 marks is for sustainability and scalability. 10 marks is for sustainability and scalability. 10 marks for presentation and communication. 5 marks for team collaboration. Total is 100 marks. So we will be evaluating all the applicants out of the 100 marks, which will go in different sectors. Is that clear, sir?
So good afternoon everyone. I will be presenting today on the topic which is ecology, regeneration potential and conservation of freshwater planarians from Pune, Maharashtra. So basically the basic understanding of planarians which we zoologists have is limited to the 9th standard textbook or whatever we have learned in the bachelor's, which just includes planaria. It is a flatworm belonging to the phylum Platyhelminthes, and it has a tremendous regeneration potential. This is what is the basic understanding that we are having. But this is not only limited to this particular things. There are vast exploration in the research potential which you can have in the planarian, and that's why it also needs the conservation. I will be explaining all these things in brief in my coming presentation. So this research study is a part of my PhD research work, as well as some research work which I have done with the Institute of Bioinformatics and Biotechnology, Pune University. Okay, so the video is not playing.
वीडियो नहीं प्ले हो रहा है, संदीप की मत करो। आपने हाइपरलिंक किया है?
Yes. Okay, so the planaria, it has tremendous regenerative potential. the other model organisms that we have been studying for regeneration, they are not able to regenerate the neuronal system or the nervous system, but it can be seen in the planaria. I will show you the images in the next coming slides, but just for the introduction, it is quite varied than the other model organisms that we have been studying. So this, even the T.H. Morgan, who is considered to be the father of modern genetics, he was once considered planaria to be the model organism for his studies, but due to the limitations in the biotechnological advances that time, he shifted his focus to the Drosophila, and we all know the rest history of it. So now the biotechnological advancements have been done, so planaria can be studied as a regeneration model organism. I will be showing you some of the things of that. So currently 120 species of planaria have been recorded globally, but in India there are only 5 species which have been documented, that too before 1983. After 1983 there is no species which has been documented in the planarians. So our study, we have discovered one species from Pashan Lake, Pune, which is Dagesia, which we have named as Dagesia punensis. We have deposited it in the National Repository of the WRC, the Western Regional Center of ZSI. With the help of the Kalavati ma'am, Samir sir, as well as the Vasudev Tripathi sir, we have deposited it. And we have also published it in the records of ZSI. It is in copywriting stage, it will get published in some days. So the first record of planaria was done as Planaria arborensis. The Richard Whitehouse, he has recorded the species and documented it in the records of ZSI journal itself in 1913. And after that, only 5 species, the Dagesia andamensis, the Bengalensis, Siamana indica, and Krishnaswamy. These are the only 5 species which are recorded from the India. And one sequence was of Dagesia from Kerala was deposited by Sola in 2022, but there is no description of the species which has been done. So the research in the planarian systematics and nomenclature is also very restricted. So it is a wide field of study open for the one who want to pursue research in it. So this is the basic systemic account. It belongs to the order Triclariida, family Dagecidae, and the genus is Dagesia. So this is the holotype and the paratype that we have submitted in the Western Regional Center. So the basic procedure, the basic protocol had been followed. For it, we have gone for the specimen preparation, DNA extraction, PCR amplification. We had to design the primers as well because there is no previous literature which is available in the Indian species, and the one which has been done on the species other than India, it doesn't apply to the one which we are having because the species is very delicate. What we find here, it is almost 3 mm to 5 mm in the length. But the ones you find abroad, they are more than 10 mm. So they are very delicate, so the protocols which apply to that species don't apply to this. So we have to standardize each and everything before performing the experiments as well. So then we went for the sequencing, and the sequence alignment and phylogenetic analysis was done using MEGA as well as the IQ3, using the 2 outgroups as media. Polychroma and Mediterranean, so these 2 were used. We used IQ3 as it is more reliable than the MEGA, and the journals also don't accept the trees built in MEGA nowadays. They go for IQ3 or Aximal. So we have done the sequencing for 3 genes, COX1, 18S and 28S, and then we went for the phylogenetics of this particular species. So this is the phylogenetic tree that we have constructed. You can see a distinct clade from here. And this is where a species stands. It is right between the species is between the other species are from Japan and China, and the one which I studied, the one which was the sequence was given by the Sola, it stands somewhere right here. So it is not related to the one species that we have found in the. So the species from Kerala that we are getting into. So this is the acknowledgement that we received from the ZSI when we submitted the species. I have also submitted its sequence to GeneBank as the publication is inline and it is not released to public yet. Okay. So the current status of the speciation is in the lentic sources as well as lotic sources in and around Pune, in the freshwater sources, you don't find planaria everywhere. You find them in the stagnant body where the eutrophication levels are very high. That's why we find the planaria. That only three different species are there. One of which we have published. The other two we have done the sequencing. We are just going for the publication for the other two species as well. So there is one which is black in color, the other is red in color, and the brown one which I showed you, the holotype and the paratype. So the brown one you find in abundance all seasons. Every time when you go for the collection, you find them in the abundant ratio. But the other two, they are very limited. If you are lucky enough, you find them sometimes. Even one unique thing which is not recorded anywhere is you can find parental care in planarians as well. So even being such lower invertebrates, they show high degree of parental care. So you can see basically what is happening. Pashan Lake is highly eutrophicated, as well as there is a lot of thermocol garbage, plastic garbage, which has been let into the water. Even the industrial effluents have been let into the water, so that's why there are eutrophication levels are very high. So they have adapted to this particular system, and you can find the planarian resting upon the plastic bottles or even the thermocol. The thermocol is the best thing for them as they are having the small cells. So what they do, they remove one ball from the thermocol layer, and there they deposit their eggs, and the other planarians, they are guarding the particular thing. So that is what I found very oftenly in the planarians. So parental care was seen in them, which was a unique thing which hasn't been documented anywhere. Okay. So I studied the population dynamics of it as well. Only 3 species are there. That's why you can see the H index, which is lying in between. It is quite low, 0.4 or 0.6. Even when I went for the Simpson index, you can find 1.9. The numbers of the planarians which I am getting is high, but the species are very less. So you can see a high number in the Simpson diversity indexes. I have studied this for 2 to 3 years now. So this is the data from 2 to 3 years. So species richness, the brown planarians you can find between 50 to 150 somewhat, but the black and red are very rare to find. You rarely find them, only after the monsoon season. You don't find them anywhere in between the seasons. You get them only after the monsoon seasons or in a little bit after the winter season. Whenever there is intermediate change which is happening, so that time you get the 2 species. So I have collected my survey sites included the Pashan Lake, or the one near the Aund river side. So also the Bugav Lake and some sources from One Garden and Yeravda near Sangamwadi as well. So I have surveyed all these collection sites, and then I came to a conclusion to have a primary collection site, which was Pashan Lake. Pashan Lake is my primary collection site. I have studied the physicochemical properties and everything from Pashan Lake. You can see high level of eutrophication. This is very low. This is after the Pune Municipal Corporation took up all the eutrophication. They do this on a regular basis and just before the rainy season, so that they just to avoid the runoff. So they do that very regularly. So in December or in January, you can see the algal blooms as well as the Econia and Pistia throughout the lake. It is very difficult to see the water as well. So this is the highest level of eutrophication. One more reason of it is there is a mortuary which is present right near the Pashan Lake. So they dip the ashes of the people as well into the Pashan Lake, so which leads into high level of sulfates and nitrites in the particular water, leading into this eutrophication levels, and which becomes a home for the planaria. So this is the correlation of the water quality parameters which I have checked for an entire year. So you can see the high levels of salinity, TDS levels, some chloride also.
Just keep.
Some chlorides and nitrates. You can see very high level of them into the water, which is a suitable environment for this particular planarians. But a unique thing about the planarians is they are not harmful to anyone. So they are not acting as a parasites. So Platyhelminths, basically we know the phylum of the parasites, but of this, the triclinids are the ones, they are not parasitic in nature. So they are not harmful, but they are a useful part in the food chain. I will come to that later. So the maintenance of culture, again after studying the physico-chemical properties, we designed the ideal culture medium for that particular planarian. The one which is given by the Sanchez and Alvarado is not working for this particular planarian, so we had to design our own culture method and for it. So this is a Montjuic water, which is particularly very costly. So rather than taking or purchasing from them or importing from them, it was not feasible for us. So we made this composition. We adjusted the pH. We have used the external workforce as well for adjusting the pH. And then we have set up the culture conditions, and the feeding is of raw liver and the hard-boiled egg yolk. Again, there was a customization in the egg yolk is not suitable for the planarians in liver, so that's why we are only feeding them with the raw liver, that too in very small chunks. Once you feed it, you have to remove it the next particular day, otherwise it gets decomposed because of the feeding. They are not such voracious feeders. So that's why they get decomposed, and by that the water parameters they get altered, and then the planarians die. They are very sensitive to the environment that they are providing them. So coming to the regenerative potential of planarian. So it is showing epimorphic as well as morpholactic regeneration. Both type of regeneration can be seen. In one type, if you just have a cut to the planaria, or if there is a wound, only that part gets developed. But if you cut planarians into very small pieces, from each and every piece you can have an entire planaria which is regenerated. So even if you have a cut near the head split, the entire head part gets developed. Intestine, if you have a cut, you get the entire intestine which is regenerating. So each and every cell from planaria is able to and has the capacity of the regeneration, which is pluripotency, which you can see in the planaria. So here you can see this is the feeding mechanism of the planaria. These are the neoclasts which we have studied. This was very preliminary study which was carried out. And you can see I had taken a cut. This is on the first day. In first 2 to 3 days, there was some regeneration happening. You can see the neoblast accumulating here, and then the entire planarian was regenerated. So same thing you can see in the other pictures as well. Even when we took a cut in between the stomach, like if we take a cut, you can see 2 different planarians heads going out from a central axis. You can see the 2-headed planarian as well. They develop 2 heads also. This was just to check the neuronal regeneration pattern in them. You can see this is the pharynx of planaria, which is an important character in the species identification as well. So this is also a defense mechanism in planaria. If the planaria gets scared, or there is some external agent which is acting on the planaria, or even for the feeding, they take the pharynx out, then they feed, and then they take the pharynx inside again. This is a defense mechanism also which is seen in the planaria. So when we went for the histological studies in the planarians, so here you can see the neoclasts. They are very abundant in the hormones and the enzymes because they require those for the regeneration. So this was the TS which was passing through the gut, and these are the neoclasts which we observed in the histological screening. So we had to develop the histological protocol as also, because of the formalin, the basic things which we use for the histological analysis, they are not working on the planaria. So formally the basic thing which we require is not working on them. So we had to design a specific fixative for this particular planarians. Even though right from the fixative up to the mountain, everything had to be standardized for this particular species. Even I had to go for cryotomy, and that things also have to be done, the BRD labeling. So the normally planarian takes 14 days to undergo complete regeneration. If it is in the normal uninduced condition, if you are not inducing it with anything, you are not going to give any growth mechanisms to it, then it takes 14 days. But if you are treating it with the tocopherol, regeneration can take place only in the 3 to 4 days. So the regeneration span lowers down to 3 to 4 days if you are inducing it with tocopherol. The mechanism behind it is tracing its roots back. So I'll trace the roots back to the cytochrome oxidase enzyme, which boosts the regenerative potential of this particular planarian. I have also treated planarian with different neurotransmitters to study their inhibitory effects as well. So epinephrine, norepinephrine, dopamine, everything was tested on planarian. So it is having, it is not boosting the regeneration, but it is restricting it. And there was one study which was carried out that the planarian hormones which planarians have, or the neurotoxins which the planarians have, are quite similar to that of the humans. So planaria can be excellent model organism to study anything of the regenerative medicine as well as the stem cell therapy can be studied in the planaria. So it can be an excellent model organism for it. So enhanced regeneration due to tocopherol, you can see the regeneration which took place. I had made the cuts to both the parts. As you can see, the head as well as the tail region both was cut. The head regenerated first, and then you can see the tail regeneration. This only occurred in 3 to 4 days. The videos are not playing, so I had included some videos as well. So this was a study which I carried out with the IBP Institute University. So I have performed molecular docking to in silico characterizations for the cytochrome C oxidase enzyme. I presented this in the Zoological Survey of India Animal Taxonomy Summit 2023, where I received Best Poster Prize for it. So this I had developed from the Autodoc and Autodoc and studied it in the Discovery Studio. I have used R software for the statistical analysis of it. You can see the 3D model protein, which is, and this study, by this study I have got an idea that the cytochrome C oxidase is responsible for the regeneration, just for the purpose of confirmation. So the neuronal regeneration, this is the part of head. You can see the neoblasts in the amputated head, and they are then responsible for the entire regeneration. You can see the neoblast accumulating at the whole site of the planarians. So planaria and its relevance now, how can we use it for the benefit of the mankind? So what is the novelty in it, and how can we use it for our benefits, whatever we are doing currently right now? Okay, so the basic thing which is happening now in the stem cell and the regenerative medicine is we have developed the organs on the chip right now. So we are not on the 2D cell culture lines now. We have moved forward to the 3D bioprinting and the 3D cell culturing. So the enzymes and the hormones which are present in the planaria, they have a tremendous effect. If we can introduce into the cell cultures of the brain cells, or even in the skin regenerating fibroblasts, we can have, it will be very better, the culture medium might be better for the surviving of the brain cells, as it is a dream which has been recognized by WHO to grow the heart as well as brain in the petri dishes. That would be easy if we just use the planaria hormones and enzymes for regenerating. Further studies need to be done on this. I am studying the 2D as well as 3D cell culture right now on planarians. I have isolated the primary cell culture as well. So this you can, from that this is the are the neoplasts under the fluorescent microscope from the primary cell culture. So why do we need to conserve the planaria? First and foremost thing is we don't know anything about planaria which is here. Right from the species identification, everything needs to be done. So if we are not conserving them, there are two things. If we don't know anything about anything, so we need to conserve that particular thing to at least get to know them. So we don't know the status of it, we don't know if they are near extinction, we don't know if they are threatened or if they are least concerned, we don't know what exactly the situation is. So for that we need a conservation. Second, for our benefit, if it is so useful in the regenerative medicine as well as the stem cell therapy, we need to conserve them. So they also are serving as a bio-indicators to the aquatic ecosystem. They are not harmful to the aquatic ecosystem as well. So okay, so they serve as an indicator to the water quality and eutrophication levels. So again, they support the food web and the food chains with interactions with the snails, insect larvae and the other which they feed upon, and the tertiary consumers such as fish from brandicula larvae which consume the planarians. So you can see a prey-predator system also, so that's why we need to conserve them. So the threats to its habitat are Pune Municipal Corporation removes Econia and Histia just right before the rainy season, which disturbs their habitat. After each removal, you can see the level of planarians. They adapt to it definitely, but the levels go down very rapidly. So you don't find them right between June up to the October, November. You don't find planarians when you go to the parkland. So this is one threat to their particular habitat. So this disturbs their microhabitats as well as the urban runoff and the pollution. If the nitrate sulfate levels go on increasing, that is also not the salinity levels after a particular threshold is also not good for them. So if the pollution is more, it is also harmful to them. If the pollution is less, again it is harmful to them. But we have to find a way to make them survive. We can construct like the things which have been done for crabs to keep them into the brackish water. So we have taken some initiatives for it. So we can also take some initiatives to preserve this particular animal. So the fast moving and disturbed waters are also unstable for their survival. So you can see the water cleaning of the water hyacinth which is being done from the Pashan Lake. So the Pashan Lake is a biodiversity hotspot, as not only for planarians, it is for other species as well. Many aquatic beetles, many snails have been discovered from the Pashan Lake. You can see many aquatic birds there. You can also see many fishes, freshwater fishes, many species of it there. So it is a biodiversity hotspot. So some conservation actions and the recommendations which we can implement. Firstly, as the place that we took right now, we should avoid the plastic contamination, plastic and the thermocol. There are some government regulatories which have been released, but we have to follow and abide to them definitely. Protect the aquatic vegetation of these particular areas pre-monsoon. Delay or reduce scale of macrophyte removal where the planarians are abandoned, but you can do something to conserve them and then you can go for the removal, as that is also very important. And ecological monitoring has to be done. So the lakes are quite neglected for the ecological monitoring, that is not being done. And raise the public and policy level awareness about the microfauna and the habitat interdependencies. So basically, understanding of the food web and the food chain is very important in the common public as well, as people have no idea about it. Killing one particular animal for the benefit of some other animal or for the benefit of some other agricultural product is not good. So basic understanding of the food chain and food web is very important. which has to be, the awareness has to be spread. So we should preserve every scrap of biodiversity as practice, while we learn to use it and come to understand what it means to humanity. So as we don't know anything about planarian, so that's why preserving of it is also very important. This is by Wilson. So thank you for listening. Any questions?
Yes, it's open for.
I have a couple of questions. As a layman or as a jury, is there any sex dimorphism? Sex dimorphism?
Yes. Actually, there's not sexual dimorphism. The male and female you can find in one particular family as well. So
They are hermaphrodites.
yes, they are hermaphrodites.
Second question, you said that you get 3 different colored planarium.
Yeah.
One is red, one is brown,
Brown.
and dark.
Yeah.
Why?
Okay, firstly, what we thought and what we read from the literature review is because of what food they are consuming. Whatever food they consume, that particular thing reflects it into a color, which is partially true, but the species is also different.
If it is so,
Yeah.
depends on the food they consume.
Yeah.
Then are they in the same vicinity?
Yeah. You can find all the 3, yeah, in the
The
same
food is actually equally available for
for all of them. They are selectively.
They are mobs.
Yeah.
Are they different mobs or?
So they belong to the 3 different species. Yeah, after sequencing we got to know. Initially we believed they belong to one species.
By now at least you must
Yeah.
be knowing that what are
They-
the different taxonomic
Yeah.
differentiations within that species.
Yes.
Or the different species. You may that you should have 3 different species in partiality.
Yeah, 3 different species in.
Are they belong to same genus?
Yes. They are belonging to Deccan.
Now, what is actually you find the role of this organism in the aquatic ecosystem?
Okay. So in the food.
It's okay, if the larger food chain and all is okay, fine.
Yes.
As you repeatedly say that they are not harmful
Yeah, they are not harmful.
to anyone.
So anyway, they are not parasitic to humans. They are not parasitic to any other freshwater species which is dwelling in the that particular area.
What about actually if they are engulfed, suppose, so the fish pond, you have to get them in bulk.
Yeah, on the roots of the planaria, when you see the roots, you can see they are not colonial or they are not a socially dwelling species, but you can find many species on the roots, dwelling on the roots of the planaria, but they are not social.
The next question is, you say that there is a parental care. So how did you observe that? Because they are on the
But now they have also adapted to our drone litter.
your lab.
No, not in the lab. In the plastic, there is so much litter which is there of the thermocol and the plastic. So they have preferred their they have preferred to live there now. So more from the roots, you don't find them on the roots. You found them in the litter. And what they have done is, as I told right now, in the thermocol there are small holes. Yeah, there are some small. They remove the walls and they keep their eggs into it. They deposit the eggs inside, and you can see many planarians which are near to it. Like if it is around, you can see them arranged.
Are they consuming? Are they consuming?
They are not consuming the eggs. No, they are not consuming the thermocol. I had studied that in the lab. I took the thermocol piece and the planaria. They are not consuming the thermocol, but they are using it as their.
One of your recommendations to protect them, you said that you should delay the removal of macrophytes.
Yeah.
This goes against the policy.
Yes, but people they are removing it very voraciously. Right from April they start removing it. So if you take some conservation steps in April, May, and then in the mid-May if you start removing, it takes more than only 2 to 3 days to remove the Econia and Pistia.
So the concept of actually the diapause like thing, you say that they are not available during a particular season.
That has to be
If
studied
they
out.
are occurrence or if they are actually sighting, they must be there. There is some or the other form of delight. So what is it? Where do they go?
That is the question which I am having. I am currently studying it. I have not studied it. Where they exactly are.
Is there any change when you study the water quality
Yeah.
or the physiochemical
Analysis.
things of the water? What happens during that period when they are not available?
Okay, the sulfur as the water.
What goes wrong for them?
Yes,
in the water?
yeah, the sulfate nitrate levels as it rains, the water gets diluted. Definitely. So the sulfate nitrate levels they also reduce significantly.
So you mean to say that actually their existence depends on the water quality?
As well as the economy and pesticide. It is all interrelated. Yeah.
The different planktons or zooplanktons, both phyto and plant. What are the associates of these planarians?
Okay, mostly the snail eggs. I found a lot of
Plankton.
in the planktons. Okay, you can find a lot of Daphnia protifers around the planarians. When
What is their food?
Daphnia.
No.
Planarians, okay, they are dependent on the microplanktons as well as they feed upon the snail eggs also. They are scavengers.
Scavengers. So what about your lab studies? What does it indicate? You must be having culture also.
Yes.
So the thing is that, sir, just a brief. So when I started this study in 2013, so in 2013 when I started this study, so I have seen so many lentic water bodies around this Pune district. And only the partial lake which I have observed, there is a lot of planarian species which I have got at near about 8 o'clock in the evening. So that time I come to the conclusion that why this planarian is observing in the night. So the first report is that the planarian is observing in the evening. When you visit to that lake at 4 o'clock, so they come to the surface of the econia, so that is we can correct. So that accidentally in 2013 I got that while screening the so many water bodies. So that planaria has been observed in the partial lake. So when we brought in the laboratory and when we start searching on that, so after having the 10 years studies on that and after comparing this species to the other, this species which we have observed in Pune region, that now we have given the name Deuterium punensis, it has a lot of regenerative capacity. And when we observe in the literature of this planarian, the developed countries like China, Barcelona and Italy, only 3 countries, they are very well studied about this planaria. No one has studied in India, only that R.H. White, that paper is 1930. And then we started doing the experiment on that planaria and we got the regeneration power is 14 days. And in other cases, the regeneration power is very high. Then we have started doing the systematic study, then it's morphological studies, and this species, it is a specific tricladed species. The tricladed species means what? This is the one intestine which has divided into the 2 parts. So it is a tricladed. And the remaining species which we want to focus, so they are not tricladed. Their intestine is completely in that. So the species which we have recorded, it has been comes in tricladed. They have 3 intestines. One is at central and it has been divided into
segregation
segregation characters also. And this is a scavenger species, sir. It is a nocturnal. It is found attached.
Nocturnal or crepuscular?
Is the dawn or evening?
Yeah.
So they avoid light.
They avoid it.
Sunlight.
Yeah. So that ethological study she has also performed on that.
The
She has
wavelength
published.
test has also been done on that.
What about they have eyes?
Yes, they have eyes. They have eyes. So ethological studies she has a chemotactic, then thermotaxis, geotaxis. So that all ethological studies she has studied on that.
What about the population of all these 3 species?
In this
Means what happens, suppose if one species is less, population, that means the other population increases?
the brown variant has been consistent. consistently more in number than the other two. But in the other two species, only after winter, and after the rainy season, the intermediate part you can find in that
There is a succession.
Yes.
Yeah.
One species replaces the other one.
Yes.
It's a succession. And sir,
But it depends upon the food availability.
Yes, the
Yeah, it depends on the
availability
food
as well
availability.
as the water parameters.
and when we got in the laboratory,
That out of these three species, they have a different level of tolerance.
yeah.
Yes, the tolerance level.
Yeah.
You can also see it in the morphology.
But food is same or all the species same?
Yes.
Yeah, food is Sandra scavenger, and in the laboratory we are giving the small chicken this liver. So and that feeding habit is again also nocturnal. When you give the food, so the pharynx comes out. The pharynx comes out and the pharynx connect the food hand. So this is
So
what.
they are such
They are
a
builder.
that
Yeah.
suck.
Yeah. Yeah. And so one important aspect of this planarian is that in foreign countries they are studying about the P53 genes. The P53 genes, it is a tumor suppressor gene in human being. So when there is a lot of cancerous growth, so that P53 genes gets activated and that suppresses the tumor. So that is the role of P53 genes. So that gene has been very well studied in the planaria in developed countries. The role of P53 genes here. Because when we cut the planaria, there is a tremendous growth of the cell. So that tremendous growth of the cell that has to lead to the cancers. But this tremendous growth of the cell here, when we cut, immediately the stem cell, we are calling it a neoblast. That neoblast start differentiating in enormous way for the regeneration purpose. But in foreign countries, what they have studied, that enormous growth of the cell, how we can block it. That they are studying in their cases. And in these species, they have a lot of regenerative capacity. So they have observed there is a PV genes in that case, that planarian species. So we are thinking that why we are not using planaria as a model organism, because Hydra is a good model organism.
Hyperplastic.
Yeah.
This species is a hyperplastic.
Hyperplastic, yeah.
And the problem is genes don't work individually. They work in combination.
Combination.
Genetics are very primitive.
Yeah.
So there is no interference.
Mm.
As far as human beings are concerned,
Yeah.
a number of genes,
Yeah.
we don't know how they work in combination.
Yeah, that mechanism.
See, we say there are 1 lakh genes in the human beings. Only 40,000 are active genes. 60% we don't know. 60,000. So it doesn't mean that 60 are not useful.
Not.
Their presence is making the 40 active. So in that case, the analogy is a little tricky.
Yeah. So we are thinking that the planaria should be explored more and it can be used as a model organism for the regenerative purpose. So here our purpose is to explore more and more planaria than knowledge. So every budding scientist, they will work on this planaria and they will explore as an Indian knowledge system. So that is our role in Indian perspective. In Indian perspective, that
Disadvantage is this size.
yeah.
Size.
So we want to explore the knowledge about planaria as a model organism in Indian perspectives. So the people from the foreign countries, they will come to India, they will study our regenerative species and they can explore it more because it is an ignored model organism because of its habitat and the habitat. And we are conserving it. That's all. Thank you. Thank you.
See, on this presentation, I have 2 questions and 1 suggestion if you want to take it positive. So you are taking about conservation of Acacia. So can you tell me what is the hackathon part of your presentation?
So this is a great platform to basically there are young researchers here, so that's why we can teach them to take this topic as well as seriously. Also, when I talked to Dr. Deccan ma'am during the ATS, so she suggested me to promote it more and more, so that more and more people can be working on this particular thing. So I found this particular platform to be a great resource for as we are dealing with the biodiversity conservation. This is one essential part of it, and also the biodiversity as well as the medical advancements, they go in hand in hand. So,
See, for any conservation,
yes.
name plays a crucial role, right?
Yes.
You said nobody has worked on this. I was expecting like, how can we speed up? Nobody is working, right? That's what
Yes,
I'm saying.
yes, yes.
How to speed up this?
But people want to explore more, more about the stem cell reasons. So we can get an awareness. There are 16 regional centers, so we can have an awareness so that people are more and more talking on this particular.
Because unless we gave a proper name,
Proper
it's
name.
very difficult.
Yes.
So coming back to your new species, what is the name you have given?
Deccan punensis.
Punensis. So yes, Pune is the name you want to give.
Yes.
Yeah, I feel there is a problem in the naming. Please consult any of the taxonomies because Article 34.2.1 of ICSL code clearly says if you are putting a noun, you can use it as, in this case what happens, if you have taken the name from, there are also species name before 2010. So where UNA is the noun.
Okay.
So to neutralize the gender, people used to put NCs. So here it is neither this way nor that way. I think if your paper is not published, I strongly recommend you should put it as Vigacia Pune and justify why you are giving. So NCs doesn't make sense here. So I think like you should consult.
They have used this old system of naming.
Yeah, that is the
Now
problem.
the amendments have come.
Yeah.
Look into it.
Once
If
the
it is
paper
not
comes
published,
out, once the paper comes out, I can change it.
yeah, then the credit goes to him.
So
The
that's
name
what I'm
will go in
telling.
bracket.
See, if I'm correct, while designing your primers, you came to our lab, right?
Yes, yes.
Our people helped us, right?
Yes, yes.
So
Even
for-
the name was suggested by some website.
So that's what I'm telling. Just asking why that. It should be either Pune plus NCs or it should be only Pune. Because if you go and check the literature, before 2001 there was a practice of putting this. I
In
also
fact,
not checked it.
actually, you are putting POO.
PUNESIS. Be aware of, see, what happens, people just follow the old traditions. Now code is giving lots of liberty to me. See, Vigacia Pune makes more sense to me rather than Vigacia Punensis. It is not Punensis, it is Punensis, right? And see, for the conservation part of view, I was expecting you will be telling something about metabarcoding or something of that sort, or like NGS thing, where, see, Sir was telling me passion. You want to know how many species of Veneria is there, just take one sip of water, do the metabarcoding. You get how many number of lineages are there. Naming is not your problem,
Yeah.
right? Naming will take, yeah.
Well, definitely.
And sir, just here, just 130 something. If you go here the technical problem is there. The planaria, even you can add planaria Kshirosagar or planaria Pune. In 2013, here I got a beautiful regeneration of this species. Means in 2013. So from last 10 years we are working on that, and I'm happy that she got funding from the government of India to work on this. So she's working extensively on that. So it's a last 10 years work. So I'm thankful that she has presented, and I'm very much thankful that the Zoological Survey of India, they have arranged a beautiful hackathon. It's a means historical moment, and I think this is the first hackathon. This is the first hackathon in 110 years, and we are the first presenter that we have studied this model organism in this hackathon. So that is our precious moment, sir. That's what we are doing. So thank you so much to Zoological Survey of India and all the referees and all the scientists coming from India. So thank you very much for giving the opportunity to present us. Thank you.
Sir, Kshirosagar sir has two students over here. One is Gayatri, other one is Mithila, and both of them are very good students. And they both are having very small child. Mithila, when last year she came here for a taxonomy workshop, she was carrying a small baby of 6 months. So I really appreciate, sir, that and both of them are having the same age kids. So I really appreciate you, sir, that you are helping these young women to take care of their family as well as take care of the science. So thank you, sir, and congratulations, Mithila, because last year I was there in the ATS and I saw her paper presentation, and everybody were just gathered near her poster. All other posters were just dried. So really congratulations to you. So now we'll go for the second presentation, and second one is Dr. Azras. Dr. Azraz from Nashik. So you're welcome, sir.
Good morning, everyone. So, the title of my presentation is Wildlife Adaptations to Human-Dominated Settlements, Implications for Implementing Conservation Strategies. So, when we talk about Ice Age, we all, the image that comes up in front of us is a woolly mammoth. When we talk about Jurassic, what we think of is dinosaurs. So similarly, in the current times, this has been officially and scientifically, categorized. This age is being called as Anthropocene, where humans are dominating the earth and all of its systems. And the extent of this dominance is so much so that it can be seen from the skies. Like satellite imageries are showing the impact we are having on the earth. So more than 75% of Earth's land surface experiences measurable levels of anthropogenic disturbances which impact wildlife community composition and animal behavior. So whatever the intact ecosystems that we had before humans spread it on the earth have been left just in bits and pieces. So the undisturbed ecosystem is just a negligible part of Earth now. Now, what exactly this means? This means that we have invaded the natural other animal spaces, other spaces humans have invaded. And how does this affect them as well as us? The interactions are likely to be altered in time and space. Because of our disturbances, animals are changing their positions, behavior in space as well as in time. There are behavioral changes, there are changes in their diet patterns and migrations due to habitat loss. We'll come to this in more detail in the next slide. Now, for example, because of human settlements, expansion and habitat loss of other species, there has been an important observation. Many species are turning towards nocturnal lifestyle or nocturnal behavior. For example, species that normally in their natural habitat, if they forage or if they hunt or if they prey during daytime, to avoid humans or to avoid human activity, they will shift their activities of foraging, hunting, preying to times when humans are not active. Now humans are diurnal animals. We are active during day and we sleep at night. That's how our biology is designed. So to capitalize on the time when humans are not active, they are shifting their behavior to nocturnal or to the evening time. Now, in this example, let's say so let's say large herbivores, they usually forage during daytime and they avoid their prey, their predators which are active at night time. But to avoid human activity or human disturbances, they have started moving towards nocturnal behavior. And this clashes with their, this exposes them to their predators which are active during night time. So this kind of disturbances we are creating in the natural world. And this has major implications that will be coming to that later. Now this figure I got from a publication recently that was published in Nature, and they have studied their detailed the shift of species from their natural timing. The implication of this, as we saw in large herbivores, the prey-predator density is being affected. studies have shown that the density of carnivores is related to availability of prey biomass, usually in natural or semi-natural ecosystems. However, we cannot distinguish any further. This part is natural and this part is human disturbance. The extent of human activities are spread to such an extension that we have to consider conservation strategies in those parts where humans have already disturbed the landscape. But recent studies have found that large carnivores can also persist in human-dominated areas by relying fully or partially on food resources associated with humans. So the earlier concept was, if there is a particular habitat where there is a high density of herbivores, you will naturally find a high density of carnivores, large carnivores. That's what everyone by default believes. But nowadays, in human settlements where there are less number of herbivores, the carnivores can still survive based on food resources related to humans. Now, we have a very prominent example of this. These pictures are very painful to watch, but how this wildlife survives in areas near human settlements. Biomass of domestic animals in human use landscape can be higher than that of wild prey. So sometimes the herbivores like deer or cheetah, the prey that normally large carnivores eat, that will be less available to them compared to carcasses, dead cows and dead meat found in human garbage to these wild animals. So they are wild carnivores. So they are likely to thrive more near human settlements. Garbage, pet food, and these waste products are leading to increase in carnivore density near human settlements. Now, a very good case study of this phenomena is leopards. Every week, almost we see 2 or 3 examples where leopards have invaded or attacked humans or cattle or livestock. This comes from a study by and Jai by 2023, a recent paper. From 2001 to 2019, the researchers reported 5,064 incidences were recorded of leopard attack. Out of these attacks, 127 attacks were on humans, 4,937 was on livestock, and leopard attacks on humans followed an increasing trend since 2012. Now, to interpret this is, since 2012, the attacks on humans has increased. So naturally, we have invaded into their space. They haven't invaded into our space. So this calls for a zone. The conservation zone needs to be strategized for the human settlements where leopard was already pre-existing, but we are now overlapping on their territory. Now this map over here shows, this case study is about Junnar region. The brown spots are all these places where the leopard attacks were recorded. Now we can see they follow a pattern. Most of the attacks were near sugarcane fields. Now sugarcane is not a natural vegetation. We had to clear forestation in order to carry out the agricultural activities. So as commonly it is understood, agriculture is an environment friendly practice. It is not. It is at par with industrialization where it can damage the environment. So again, to proceed further with this leopard case, it has been recorded that complete dependence of leopards on domestic animals as prey in croplands of Ahmednagar district of Maharashtra, India. Again, this paper was done by Atreya, and she has got a lot of extensive work on leopard-human conflict. They analyzed around 55 leopard scats. Scats means the droppings, leopard droppings. And they found 87% of the leopard's prey biomass consisted of domestic animals. So this includes cattle, livestock, basically. And out of this 87, again, 37% of this consisted of domestic dogs. Now, if you search on YouTube or if you look at the newspaper clippings, it's very common for leopards to feed on stray dogs. In fact, farmers have this what do I say, a different strategy where they keep pet dogs in order to it's like the first defense system against leopards. So the leopard will attack the dog, and that the attack is diverted. So point being, animals are adapting to human settlements or human behavior. Another example I could cite from the talk before me, where planaria are making use of thermocol as a habitat. So in wildlife, all around us, animals are adjusting to the Anthropocene changes made by humans. Discarded another anecdotal incident that I heard from a forest officer. He said, what do I say? The chicken transporters, they usually throw the dead chicken, in abandoned places or far off moors, and those dead chickens are again attracting leopards. So again, we are seeing a transfer, a shift from predator food habit to a scavenger food habit in leopards. So the point I wish to arrive at is the conservation efforts need to be considered not only to the pristine, untouched old forest, but also to the human use or mixed use landscapes where human settlements are very prominent. Considering the extent of human invasion in the natural habitats of wildlife, a significant territorial overlap is to be expected with humans and wildlife. So human-wildlife conflict is going to increase, we like it or not, the way we are invading human spaces. Hence, conservation efforts and strategies need to be implemented not only in protected areas, that is sanctuaries and forest areas or national parks, but also in human-use landscapes and disturbed forest areas. Now, how do we go about this? I found this excellent paper where they have developed a theoretical model. Very often we hear about elephants or wild boars, they damage agricultural crops. And this is a very disturbing scenario which has direct implications on economics. The agricultural distress is a huge thing in Maharashtra as well as in India. Now on top of it, damage by wildlife is going to further aggravate the situation. Now this paper deals with how do we handle or how do we mitigate the damages caused by wild animals to agricultural produce. Now damage to agricultural crops by wild animals is a natural phenomenon. It has been happening since centuries, right from where we start, maybe when agriculture was discovered. However, the increase in human population, this scenario is more acute now. Now, from the current reports, farmers are never satisfied with the compensation that they get against the damage caused by wild animals. Now this makes a major liability for conservation efforts. Farmers or tribal people who live around the protected areas of national parks and sanctuaries traditionally are animal loving people. Their traditions and culture usually promotes worshipping and protecting animals. However, when there is the authority that takes care of these animals, there is a psychological mindset that kicks in that these animals are government's responsibility. It's no more a common social responsibility. Then this mindset promotes the expectation that if there is any damage, government authorities or NGOs or whichever, someone else is responsible for those animals, and they should be compensating us for the damages that are caused. Now, working out these compensation plans or policies is very difficult. So far, we have had a very disappointing performance on this front. Many times, farmers face genuine economic loss because of wildlife damage. However, the compensations that they get are meager. They are not enough, quite disappointing. And this leads to retaliatory action from the community that lives around the protected areas. This leads to trapping animals, hunting them down, poisoning them, or electrocuting them so far. Which again, there are studies which show that culling or lethal retaliatory action against wildlife damaging.
Sir, just 2 minutes.
Sure.
2 minutes I just want to interfere. किसी का vehicle number है MH12 XZ3514. कार MH12 XZ3514. किसी का भी नहीं आपने से? So thank you.
So, to avoid these retaliatory actions or unplanned management, that could harm the wildlife. The authors pointed out some of the difficulties in formulating a good policy when it comes to designing a compensation package for the farmers. Free from corruption, the assessment that is done for the damaged crops is usually not very honest. The authorities or the people at the authority position may not always be inclined to report it honestly. There could be a favorism, there could be a negative bias, or there could be a positive bias in order to carry out the assessment of the damage caused by the wildlife. Again, fairness of the compensation. Farmers are likely to be undercompensated or overcompensated. Undercompensation is always not desirable because that will again lead to retaliatory action. And overcompensation is also not desirable because that may encourage others to settle in the nearby areas with the expectation that they are going to get more money out of government for the damages caused. Another problem is no free lunch. Many times damages are not genuine, or maybe the damages are not caused by the wildlife. Damages could be the conventional pest or some other negligence from the farmer side, which may be unfairly or incorrectly documented as wildlife damage and may lead to policy collapse. In long term, such incorrect documentation always leads to collapse of the policies are not implemented. Another problem is assessment requires extensive manpower. So a policy should be designed in such a way that it may not be an extensive exercise to assess the whole kind of damage that was caused by the wildlife. Another, as I mentioned, avoid your animal syndrome. The farmers get under the impression that the wildlife is government's responsibility. So it is government's job to compensate us. Many times we may not take, it's again a human nature. The moral responsibility might go away in pursuing the livelihood that it is them who has invaded into the natural territory. So again, this is another major reason for causing that psychological divide, where the victim is the farmer and the compensator is the government. So that psychological divide also needs to be handled by developing this policy. And sensitive to changing ecology. The damages that are caused by wildlife animals are dynamic. It's not a very steady thing. So animal populations change, densities change, habituation of human presence, preferred crop species, and market values. More importantly, what was the current market value at that point of time? So these are the various difficulties that are that need to be taken care of when implementing such policy. Now the authors here those were the difficulties and what needs to be further considered while developing this. In agricultural economics, it is the net produce that is important. So I'll cite an example. Let's say there's a rice crop, there's a rice plantation at a particular area, and the damage was caused quite early in the plantation. Like the saplings was quite small, and that's when the damage was caused. However, there is a chance that this damage is likely to be recovered, in spite of the invasion of the boar or whichever wild species damaged it. So it's during the assessment, if we just assess that this has been damaged and if we calculate this much compensation is to be made to the farmer, it won't be fair to the policymakers or the farmer. So the net produce is important. I'll come to this, why this is, how this will be considered, how the authors have considered this while formulating the formula. Another, to implement the scheme, they propose some steps. Now, we need to make guilds of farmers or the communities that live around these protected areas. These areas is a zone, the buffer zone, where the wild natural habitat and human habitats are overlapping. So let's say, for example, there is a national park, and 1 kilometer area around the national park can be zoned as Zone A. And this Zone A is likely to have most exposure to wildlife invasion. 2 to 3 kilometers area around the national park can be considered as Zone B. So this area is likely to have a lesser exposure or damage caused by wildlife. So a group of farmers adjoining the protected wildlife area who grow the same crop and share a comparable risk of depreciation. Farmers exposed to comparable risk from form a cooperative group. As I mentioned, the 1 kilometer zone around the park can be clubbed into one cooperative group of farmers who share the same risk of wildlife damage. Now, out of the N farms, let's say we consider 5 to 6 farmers and their farms into one group, would be carefully fenced to prevent entry of protected crop raider species. Now, this fencing, this is a precautionary measure. This would depend on the type of damage or the type of species that is, type of crop or the type of species that is going to damage the crop. Now, there are elephants, there are wild boars, there are gores, there are nilgais in MP. So, depending on the animal, the few of the farms will be well protected and well guarded. So that could be wired fences or electric fences or a kind of protection that are non-lethal to the wildlife. These will be the control group of the farms that are being protected. And the remaining farmers will be taking their own precautionary case based on their ability, because they will have to divert their resources from their farming practice towards, they will be needing additional resources, additional investment to make those precautionary protective measures. So it would depend on each and individual's capacity. However, 2 to 3 farms will be well protected up to as per the recommended measures. This could be undertaken by the forest department or the concerned authority. All other farmers will leave their farms unfenced but are allowed to guard them by non-destructive means such as shouting, chasing of animals, care device, etc. But destructive means such as firearms, traps or poisons will be strictly banned. The remaining farms. I'm talking about the group that we have selected, the most exposed group, let's say group A. 2 or 3 farms will be well protected. Now that can be done by the farmer himself or by the authorities. Now this is going to add up to the authorities' operational cost, but that's what the purpose, the budget allocation is for the forest department. So the purpose of this expenditure will be explained further again in the next slide. Now once this preparation is done, the mathematical model for this compensation would be, after collecting the harvest data, after the season is complete, the yield is done, all the farm groups, the compensation will be calculated as X average yield per unit area of the fenced controlled farms. 2 or 3 farms that were fenced by the authority, under the authorities' guidance, their average yield will be calculated. Again, the average yield per unit area of unfenced farms, and YI, the ith farmer yield. Now, the compensation for the ith farmer will be average of the fenced control minus average of the unfenced farms divided by the average unfenced farms multiplied by that particular farmer for whom we are calculating the compensation package. The compensation package is designed using some of the principles of the mathematical theory in behavioral economics. This is called as game theory. There was a very popular movie some years ago, Beautiful Mind. This scientist who developed game theory won a Nobel Award for it. Now, game theory assumes that all the participants in this mathematical model are selfish. They behave or they operate for their self-benefit. They do not have any altruism. So, based on this game theory, this formula was developed, and again we'll come up to the further assumptions how this calculation will be fair for everyone, in spite of someone trying to play the system. If someone tries to cheat by overestimating or underestimating their loss. So this data was not just theoretical. They also implemented this in some test ground truthing. Some ground truthing was done for this formula, and most of the findings were applicable or were aligned with the model that they ran. Now, for example, in 2013 and 14 and 14 and 15, an experimental comparison of yields in neighboring fenced and non-fenced, non-guarded areas was done as one of the methods of assessing the extent of damage. Data from the fenced area can be used as the protective control, that is X average, for the model. For rice in 2013, the control yield was 26.07 quintal per hectare, and the average of 9 other farmers was 15.67 quintal per hectare. So, if I may work out this, the fenced farm were protected from the wildlife damage. No wildlife could damage those farms, and those farms gave the produce of 26.07 quintal per hectare. But the remaining farms were unfenced. Those gave the that were 9 other farms average. Those gave 15.67 quintal per hectare. Now it's a pure statistical thing. A wildlife damage is totally random. They may not target one particular farm. So the average of unfenced farm is always going to be low compared to the control farm. But the fenced farms give us the maximum yield that can be possible from that field. And that can be the threshold that can be used to calculate the compensation that be given to the farmers who have suffered the damages caused by wildlife. As a result, the percentage compensation was calculated to be 66.33% for over each farmer's yield. So this 66.33% is being calculated by the control farms, which give us the estimate of maximum yield that could have been gained from the fenced farm. And by the current market value, the average compensation due per farmer was 17,523. However, for that particular year, the compensation that the farmers got was just meager 4,000 something, 4,000 some change. So obviously, this is going to aggravate the situation. Farming population is going to be angry with the government authorities as well as with the wildlife that is damaging their livelihood. So that's going to further complicate the situation. Implementing or